Hi, everybody. This is Dr. Launderville. This is your third podcast. And continuing on our walk through cell signaling, we're going to get into some more pathways. And there are many classes of signaling pathways that we will talk about. We've talked about how there's this receptor-associated kinase. We've already talked about some of these where a kinase gets activated. We talked about protein kinase A last time. And that kinase traveled to, the phosphorylated product traveled to the nucleus. We talked about GPCRs. Later on, we'll talk about Wnt, hedgehog, and NFK beta, and Notch and delta we're not going to talk about. But the theme of something arriving on the outside of the cell, changing what gets transcribed as a result of that thing arriving, we're going to continue with that theme. How does that happen? Well, here's an example where you had two forms of the receptor. They come together and they dimerize. That activates a kinase on the inside of the cell. There's an exchange of GDP for GTP, and then there's what's called a kinase cascade. We'll go over that in this podcast. Then that transcription factor that gets activated by phosphorylation goes through the nuclear pore, and it's going to unwind some of your DNA on these nucleosomes such that it can be transcribed, and goes from closed chromatin to open chromatin, and then these genes can be transcribed. So we've already used the term cytokine, and I talked about in the COVID-19 podcast a cytokine storm. The term gets thrown around a lot, and I just want to make sure that everybody understands what it is. So a cytokine is a protein, okay? And it's a protein found outside of cells. So you don't find cytokines inside cells, and you don't find cytokines that are not proteins. So, and they are small. They are proteins that circulate in the blood, so outside the cell. They're released from one cell and affect growth and differentiation of other cells. So in COVID-19 disease, white blood cells that are caused to the site of cell damage from viruses, they release cytokines which are circulated in the blood, and they call other cells to that area. There's huge diversity in cytokines. They are all evolutionarily related to each other. They have this 4-alpha helix quaternary structure or tertiary structure if they're not multi-subunit. So again, all cytokines are proteins. Only cytokines are found only outside the cell. Now hormones can be proteins, but they don't have to be proteins. So there are hormones that are non-protein. Some are lipids. They can have any kind of secondary structure if it happens to be a protein. And they're more typically secreted from a particular tissue. So like an adrenal gland, for example. That's not always the case. Some hormones are secreted from a lot of tissues. And sometimes things that we thought were only secreted from one tissue found out later, years later, that they're secreted from multiple tissues. But again, hormones can be proteins or non-protein. They tend to come from one tissue, and they may travel through the blood like a cytokine, but they tend to come from a particular tissue. I've seen both terms used interchangeably for some proteins. So leptin is a protein that I studied. There's also a hormone. They talk about them being peptide hormones or protein hormones. And certainly you can be both a protein and a hormone. There are some hormones that are definitely not proteins, however, like testosterone, for example. Okay. There are many receptors that we will talk about that are membrane bound and they have kinase activity. We'll talk about some today. So there are things called receptor tyrosine kinases that are receptors. They have a tyrosine residue on them that gets phosphorylated as part of them being activated and becoming kinases. So RTKs are receptor tyrosine kinases. This whole structure is one polypeptide chain that's folded up into this structure, has an extracellular domain and an intracellular domain. And as part of that single polypeptide chain, you have a domain that acts as a kinase. Now you can see the same shape in another situation where the kinase part is a separate protein, a separate polypeptide chain. But in the case of RTKs, it's all one polypeptide chain. So these are activated by binding a ligand that causes them to dimerize. It could be the stoichiometry of 2 ligands per 2 receptors or 1 ligand per 2 receptors. But the receptors dimerize. When they do dimerize, there's a phenomenon called autophosphorylation, where the protein phosphorylates itself using ATP as a substrate, attaches a phosphate to itself, the tyrosine residues of that, and then once the domain that is the kinase gets activated by being phosphorylated, it in turn phosphorylates many other residues on the intracellular domain of that RTK, receptor tyrosine kinase. Okay? All coded for by one gene, one primary amino acid sequence codes for the whole thing. And that's an important distinction that we'll talk about throughout the rest of the course. So again, dimerization is key. This is a receptor tyrosine kinase, the epidermal growth factor receptor. It has extracellular domain. Here's the first messenger that's floating around the blood, epidermal growth factor. 2 copies of epidermal growth factor bring together 2 copies of the receptor, causes it to dimerize, causes the intracellular domain to be activated. And once it's activated, multiple phosphorylation sites will get phosphorylated on itself. So it's called autophosphorylation. And these are typically tyrosines. Sometimes, if it's an RTK, it's a tyrosine. Sometimes in other similar situations, you would also see serine get phosphorylated, and typically the actual amino acid number is well known. So tyrosine number 329, for example. And in fact, you can even buy antibodies against a particular amino acid once it gets phosphorylated, because it turns out that they're not all equal in terms of how they cause things to happen in the cell. Which amino acid gets phosphorylated can cause different things to happen. Here's another example of a RTK receptor. This one's really famous. This receptor is called HER, H-E-R. And it comes in many different types of forms. These are all evolutionarily related to each other. They have different amino acid sequence, but the sequence is similar enough and the structure is similar enough that we know that these are evolutionarily related to each other, just different versions of the same thing. So there's a HER1, HER2, HER3, HER4. They tend to have different ligands that bind to them, and they have different tissue expression, etc. Typically, in normal signaling, you would have 2 copies of HER1 dimerizing, or you could have a HER1 and a HER2, HER3 and a HER2, HER4 and a HER2, etc. This is a a situation that happens in about 1/4 of breast cancer patients, where the HER2 is overexpressed and also forms heterodimers with other HER isoforms. And then once it's in a heterodimer, it is more sensitive to epidermal growth factor stimulation than it would be as, for example, in a homodimer. So this one here is more sensitive to epidermal growth factor stimulation than this one is. And just like its name says, it's a growth factor. And so it causes cells to get a growth signal. In other words, go ahead and progress through mitosis and duplicate the cell more often than should be. And so you have uncontrolled cell growth or cancer. One strategy that has been really successful against HER2 positive cancers, and when we get to talking about cancer toward the end of this semester, one of the things that distinguishes different types of cancer is their phenotype, or what kinds of proteins are they expressing. So if you've ever known someone who has breast cancer, you'll know that they get typed as to what kind of breast cancer they have. So it's HER2 positive, estrogen sensitive, that sort of thing. So if a person is HER2 positive, there are monoclonal antibodies that are given as a drug, a biologic drug, and they can identify these tumors because they're overexpressing HER2. And once antibodies identify cells, that is a signal to white blood cells to destroy those cells. So that's a chemotherapy that doesn't have all the side effects of other chemotherapies like hair loss and nausea and that sort of thing, and can be really effective at eliminating the tumor or at least shrinking the tumor and reducing the chance of recurrence of that tumor. So HERs are examples of RTKs. Once you have bound your ligand and you've dimerized and you did the autophosphorylation thing, there are proteins that form a interaction between the bound protein, the phosphorylated protein, which has a different shape, and the ultimate downstream effect. These are some examples of these proteins: SH2, SH3, etc. And SOS. These proteins are adapters that will ultimately interact with RAS. Here's RAS. So we talked about RAS, remember, in the last podcast. And RAS is another signaling molecule that's going to switch out GDP for GTP and become activated. RAS doesn't bind directly with these RTKs, but it can through these adapter proteins. So it's as if RAS needs a 3-prong plug, and this is an adapter if you have a 2-prong plug, or the other way around. If this is the 3-prong plug, these are the adapter proteins that allows RAS which is a 2-prong plug, to interact. Here's just an example of an SH3 domain, which is an adapter protein, and how it interacts with the SH2 in this case domain. So how those SH2 and SH3 interact in a space-filling model. Okay. So you bound your ligand, you dimerized, you did the autophosphorylation thing, you recruited your adapter proteins, and now you've activated RAS. Well, what does that do? Well, RAS activates what's called a kinase cascade. So one kinase gets turned on, activates another kinase, and eventually something that is phosphorylated will go to the nucleus, to that open chromatin, and cause something to be transcribed that wasn't transcribed before this happened. So if we go back here and we say, okay, we recruited our adapter proteins and now we've turned on RAS, and now we have active RAS, what happens? So inactive RAS becomes active RAS. RAS can activate RAF, which is a kinase. So normally RAF is kept inactive by binding of a protein called 1433 that binds to phosphorylated residues on RAF, keeping it from being an active kinase. RAS will knock one of those 1433 residues off so that now RAF can be active, and it's going to autophosphorylate. It's phosphorylating itself, and then also it's going to act as a kinase to phosphorylate MEK, and MEK is going to phosphorylate itself, and then it's going to act as a kinase to phosphorylate MAPK, and then MAPK goes to the nucleus, activates lots of transcription factors that need to be phosphorylated in order to be activated, and you change what gets made as a result of that. So this is a part of a kinase cascade that really started back at the receptor. When the receptor bound its ligand, it autophosphorylated, then it recruited the adapter proteins, then it activated RAS. RAS activates RAF, RAF activates MEK, MEK activates MAPK, MAPK substrates go to the nucleus and change what gets made. You're going to see kinase cascades again. You'll in particular see this protein 1433 again in a couple other places. Okay, what's the difference between an active and an inactive MAP kinase? There's this thing called an activation loop. Involves this threonine, this tyrosine amino acid number 185, threonine amino acid number 183. They change position when it's activated, and they get phosphorylated, and that changes the shape of the protein. This activated loop becomes active, and then it can autophosphorylate and act to phosphorylate other things. Okay, so here's first question for the H5P. Okay, so we've talked about kinases since the beginning of the semester, and we talked about a bunch more in these online lectures, so you can be sick of hearing about these kinases. And lots of people were sick about hearing kinases. So when this one was discovered, it was called just another kinase, or JAK, J-A-K. For short. Sometimes referred to as Janus kinase, because Janus, I think Janus was a Roman god. Anyway, a two-faced god, and this protein has two faces, one that is active, one is not active. I much prefer the just another kinase story. So JAK kinases are super famous. They're in all kinds of pathways. JAK kinase is activated in COVID-19 for interleukin signaling in the early phase of that disease. It's similar to RTK. In fact, it looks really similar to RTK, except the kinase part, okay, and the receptor portion are coded for by different genes, okay? So in RTK, back here, all of this thing is one polypeptide sequence that folds up into that. For JAK kinases, this thing here, that is the kinase, is a separate polypeptide sequence coded for by a separate gene, then the receptor part, okay? And in this case, they show you one ligand binding to two receptors, and they dimerize and it becomes active. That's not always the case. It could be two ligands binding to two. The stoichiometry is different in different kinds of JAKs. And you'll hear a lot about different JAKs, JAK2, JAK3, JAK4, JAK5 in different situations. But it's similar to what you've already seen in that you have dimerization, phosphorylates itself, and then phosphorylates, in this case, the receptor part. So it's not really autophosphorylation in this sense, in that first the kinase gets activated and then it phosphorylates things that are nearby, but it's not really the self because these two things are coded for by separate genes. But again, tyrosine is the key. So JAKs signal through STATs, and STATs stand for signal transducers and activators of transcription, STAT. And the STAT is going to bind to the DNA directly and change transcription activity for a particular gene. So once, this is erythropoietin, for example, it's a ligand that causes JAK receptors to dimerize, activates the kinase. Once the kinase gets activated, phosphorylate multiple residues on the EPO receptor. That changes the shape of the EPO receptor, allows the STAT protein to bind. It has an SH2 domain, very similar to what we saw before in the RAS kinase cascade, and a DNA binding domain. Okay, these STATs, once they get phosphorylated by the activity of JAK kinase, will dimerize, and then they will go to the nucleus. And this is a more realistic picture of what a STAT complex looks like when it's bound to DNA down the middle, and the STAT complex surrounding the DNA, and binding to open chromatin, and again changing what gets made as a result of that. So there are lots of JAKs, at least 5 different families of JAKs, and also lots of STATs, STAT1, STAT2, STAT3, so on. And you can have different types of JAKs interacting with different types of STATs. So it's not that JAK3 always interacts with STAT3. It could be JAK2 and STAT3 and so on. EPO is a cytokine. Okay, so I said cytokine. So I know it's a protein. I know it's small, and I know it's circulated in the blood. Okay, and it's a hormone. Okay, so EPO is a small proteinaceous hormone circulated in the blood, cytokine, signals through JAK-STAT pathways, and what genes get turned on as a result of those STATs going to the nucleus and binding to the DNA are genes that increase red blood cell production. Amgen, which is a biotech company, they produce synthetic EPO, and they use it as a drug for patients who have low hematocrit. So someone who's going through chemotherapy might have a low hematocrit. Hematocrit is just the percent of your whole blood that is red blood cells. So if your hematocrit is 32, then 32% of your blood by volume is made up of red blood cells, so on. Red blood cells are the things that carry oxygen. And so increased hematocrit can lead to increased performance of your muscles because you're supplying your muscles with oxygen. You can't go too far because your blood literally gets thick, gets sludgy, because there's so many cells in it. This is one of the drugs that Lance Armstrong used and denied using, and he got caught using it. And it's sort of definitely a hard thing to catch, because you make EPO. Humans make EPO. And if you do a drug test for somebody and they have unusually high EPO, then you can say, hey, what's up? Why do you have such high EPO? And they can say something like, well, that's what makes me a professional athlete, is I'm genetically superior in terms of I'm making more EPO, and that's why I have such high hematocrit. However, they finally found him with mass spec analysis, in that the synthetic EPO has a very particular mass spec signature, a very particular molecular weight down to the 10th of a Dalton. And it's very unusual for naturally made EPO, the EPO that you're making, your body's making yourself, to always have exactly that size EPO down to the 10th of a Dalton. And that's finally one of the things how he got busted. Okay. Another JAK-STAT signaling protein hormone pathway is leptin. Leptin is the thing that I study. I don't want to just bowl you over with it, but it's pretty cool stuff, if I do say so myself. And it is something that you're going to hear a lot about in professional school, grad school, PA school, vet school, med school, because it involves all kinds of physiological systems, and so it's worth talking about. And leptin signals through JAK-STAT receptors. So it was discovered in 1994. These mice have been around since the 50s. This is a wild-type mouse. This is what mice normally look like. And this is what's called the obese mouse, or the OB mouse. So OB means has a homozygous mutation in the obesity gene. But they didn't know what the obesity gene was for a very long time, until Jeff Friedman's lab at Rockefeller University cloned the gene that caused this sort of phenotype. Before they knew the genetic basis for this obese phenotype, they would do these sort of Frankenstein sort of experiments called parabiosis, where they would take the blood supply of this mouse and hook it up to this mouse. So blood would leave this mouse, travel through this mouse, and then come back into this mouse. Again, that's called parabiosis. And then when they would do that, this mouse would get thinner and this mouse would not get fatter. So they knew that something was circulating in the wild-type mouse, and that was missing in the obese mouse. And it took a long time to find it, discovered in 1994. And the first model out there for how they think leptin works, and this is probably the one that you're going to run into most often, even though it doesn't really hold water anymore, is the idea that leptin is made by fat tissue. Okay? And so adipose tissue, the fat tissue, is the thing that is making this hormone and secreting it into the blood. So if a mammal has a high concentration of fat tissue on their body, then they will have high circulating leptin cytokine in their blood. The receptor that reacts to leptin, the leptin receptor, which is a JAK-STAT receptor, has high expression in the brain, in the hypothalamus of the brain, where it binds and activates JAK-STAT pathways. Those pathways will turn on STAT genes that will bind to open chromatin and turn on expression of genes that stimulate metabolism and at the same time reduce appetite. So if you are stimulating metabolism, you're burning more fat, and at the same time you're taking in fewer calories, that will lead to lower fat because it's got nowhere to go. If fat is the thing that's making leptin, then lower fat means that now lower amounts of leptin are circulating in your blood. Eventually it falls below the binding constant for those JAK-STAT receptors in your brain. It'll fall off that receptor in your brain. It'll undimerize, it'll monomerize, and therefore you're no longer phosphorylating those stats, no longer transcribing those transcribing those genes that lead to increased metabolism, so your metabolism reduces and your appetite is stimulated. So this sort of back and forth is called an adipostat. You can think of it like a thermostat. So thermostat in your apartment or your house is set at a given temperature. If temperature goes too high, the furnace goes off. If temperature is too low, the furnace comes on. So if your fat stores are too high, you'll stimulate things that will burn that fat. If your fat stores are too low, you'll stimulate things that will cause the fat to increase in concentration. And this is an appealing idea because most people do have a pretty tight tolerance in how much body fat they have on them over the long period. So an example is if you drank an extra 4 ounces, which is a little tiny cup of orange juice every day, over the course of a year, that would turn into 10 pounds of extra fat. So if you're just having that many more calories than you need every day over the course of a year, that's 10 pounds. And most people don't gain that much weight over 10 pounds. So the balance of how many calories we're taking in versus how many calories we're expending is actually pretty good for most healthy people. So soon after this is discovered, lots and lots of studies are done because of cha-ching, right? So there's a lot of the American public is obese. It's about 1 in 3 now. And so if there was a pharmacological way to alleviate that, any drug company can make lots of money. So lots of effort went into figuring out how leptin works. And by lots of effort, I mean something like over 50,000 studies in the last 20 years. So some of the things that we know in all that work. We know that leptin, how much is circulating in the blood, and how much body mass is made up of fat are positively correlated. Now, there's a big cloud of numbers behind these lines, but it's definitely, there is a trend there, and it's a significant trend, okay? It doesn't explain everything. It only explains, this is an R value, so it only explains about, say, 30 to 40% of the variability, but still, they're correlated. It does a lot of other things too. So these are data from the original papers describing leptin, and ++ means wild type mouse. OB is the mice that have the mutation. And in the mice that have the mutation, they have a lower metabolic rate. That's what this is here. And if you give them a shot of leptin, their metabolic rate significantly increases. Okay, and these are heterozygotes. Not only just metabolic rate, but also body temperature. They have a low body temperature, these OB mice, and their body temperature normalizes if you give them a shot of leptin. So like I said, 50,000 studies. What else does it do? It's more like, what doesn't it do? I've read many leptin studies, but I still get surprised all the time when somebody tells me, and it does this thing too. And I shouldn't be surprised anymore because it seems to do everything. But some of the highlights, if a mammal is starving, it will tend to have a compromised immune function. And if you give a mouse or a rat that is starving injections of leptin, their immune function recovers even though they're still starving. So even though they aren't getting any more calories, leptin will rescue that function. It affects bone density in a very complicated way. So it seems to act differently in the OB mouse than it does in a wild type mouse. Really high leptin can inhibit the activity of bone building cells, and really low leptin mice, these OB mice, will have thicker and shorter bones. But I'll talk more about that. It's more complex than that. There is an interesting effect on reproduction, and that very low body fat mice have low levels of leptin because leptin is made by body fat. But they also don't enter puberty. This can be reversed by giving them leptin injections. This also happens in humans. So there's very few people who have leptin mutations. I think there's only one family that's been identified. And people in that family were grossly obese. They also never entered puberty into their 30s. They were put on recombinant leptin therapy, and they regained sexual function or initiated sexual development. It can also cause capillaries to grow and in general cells to grow. So more on the bone thing, just because it is complicated. There's about 100 or so papers that very strongly say that the effect of leptin on bone is to build bone. There's also about 100 papers that say very strongly and with good evidence that the effect of leptin on bone is to destroy bone. So what's going on here? First, they thought that the difference between these two studies is how the leptin was delivered. So one way is to give it interperitoneally, so into the peritoneum, into the body cavity. Another way is to deliver it ICV, or intracerebrovascular, so into the ventricles of the brain. And you get different effects with different delivery methods. So they've done experiments where they use OB mice, so they can't make leptin, and then they use a recombinant virus to deliver the gene to make leptin, and they can do that just to make leptin in the central nervous system, but no leptin actually circulating in the blood. And when they do that, it has an effect on the bones. So this is the size of a femur of a mouse that's been untreated. If it's an OB mouse, they tend to be shorter. And then if you replace leptin production just in their brain, so nothing circulating, their bones recover their length. So they said, okay, well, that's it. But then it turns out that that's not the end of the story. It's never the end of the story. There have been studies in the last 3 years or so saying that people who have had gastric bypass surgery to dramatically lower their body weight. So they have their body weight or something like that. Then they follow them for a couple of years and they look at their bones, and their bones are dramatically decreased in bone density after having this surgery. So much lower leptin signal and decreased in bone density. Okay. So what does that mean? I don't know. I don't know what it means. But here's a hypothesis about what's going on with bone. And this is something that I've recently proposed to the National Institutes of Health, so don't steal my idea. Okay. So I think what's going on is that when you are growing, leptin stimulates bone growth because you're sensitive to leptin when you're growing. You have lots of receptors, and lots of that JAK-STAT pathway is going on. And these OB mice, they have never had leptin signaling. You give them a little bit of leptin and they're very sensitive to it, and they have a big reaction to it. But when you become an adult, you are less sensitive to leptin. Less leptin receptors are present on your cells, and so the effect of a leptin dose is to remodel your bone, and just to, switch out bone like normal bone tissue does, and constantly building and remodeling that bone so it stays healthy. But it doesn't have big effects on bone as an adult. So if somebody had a big dose of leptin as an adult, it wouldn't have much of an effect because adults are less sensitive to leptin. That is unless you break a bone, and in that case, leptin definitely stimulates bone growth. They've done this in wild-type mice over and over. At the wound site, you get increased bone growth with leptin. By the way, I just looked up baby clip art and I got this, and then I said, give me a clip art of an adult, and I got this. So as adults, I guess all of us have looked forward to lawn mowing, as that's what it means to be an adult. Okay, here's another question for H5P. All right, so that's on the leptin side of it. On the receptor side of it, remember it's a JAK-STAT receptor, and there's lots of different kinds of leptin receptors. This is a diagram where TM stands for the transmembrane area, in black box there, that's where it crosses the membrane. And then this is the part that's inside the membrane, this is the part that's outside the membrane. So this would be facing the blood, facing inside the membrane. All these different kinds of receptors, really this is the only one that we know much about. This is so-called the long form, the OBRB, and this is in that model I told you about, the adipostat model, that's talking about this kind of receptor. What these other receptors are doing, most of them haven't been studied much. There is some evidence that this thing here, which doesn't have any transmembrane domain and is soluble, can bind leptin in the blood, and it might control how leptin is transported across the blood-brain barrier. Because how much leptin you have circulating in your cerebrospinal fluid is really the amount of leptin that is going to see the receptors in your brain, that's going to affect your appetite and all that sort of stuff. And there are a lot of people thinking that that is the key step. It's just getting it into your brain from your circulation. So there's about 10,000 papers, literally, you can read on that. All right, maybe 2,000. Okay, so I mentioned before my hypothesis that about what I think is going on with leptin in bone is that there's a change in sensitivity to leptin. Well, how do you get a change in sensitivity to leptin? This is a well-known phenomenon in other signaling systems, like in insulin, for example. So one of the causes of adult onset diabetes is fewer insulin receptors on the surface of cells. And so it takes more and more insulin to do the same job as it did when you were more sensitive to insulin. So a similar thing may be happening in obesity in humans, in that there's a big leptin signal for a long time coming from fat tissue, and the cells are responding by pulling receptors off the surface. And so you become less and less sensitive to leptin over time if you're obese, and therefore it takes more and more leptin to do the same job as it did before you were obese. So there's this thing called OBR-GRP. It's also known as LEPROT. So OBR-GRP was the obese receptor gene-related product, and LEPROT was leptin receptor overlapping transcript. These are genes that are coded for by a frame shift within the leptin receptor gene. So you have the leptin receptor gene, you have a start codon, in that leptin receptor gene, and you go away and you make one of those versions of leptin receptor. But there's another start codon that is out of frame, so not using the same codons as the leptin receptor, and it codes for a totally different thing inside the leptin receptor gene, and that thing is now called endospannin. And when this was knocked out in mice, it turned out that you could not make these mice obese. So you put them on the couch, feed them Doritos all day, they munch and munch and munch, and they never get overweight. So this thing, now called endospannin, governs traffic of leptin receptors as they're coming up through the Golgi, going to the cell surface. And if you make a lot of OBR-GRP, you keep your receptors underneath the surface of the cell, and you only put a couple of receptors out there. But if you get rid of OBR-GRP, then all your receptors go to the cell surface because that's the default pathway. Now you're super sensitive to leptin, and you turn on leptin signaling if there's any kind of leptin around, and leptin signaling results in turning on fat metabolism genes. So these guys ramp up their fat metabolism when the endospannin goes away. All right. So that's a cool little bit of molecular biology about leptin. There are other cool little bits in the leptin story. And one was that it was really hard to find a non-mammal version of leptin. I mean, like 15 years hard. Maybe it was, maybe 10 years hard. Anyway, the normally how you would do this sort of thing is what we talked about when we talked about gene cloning. And when we talked about gene cloning, we said you line up a whole bunch of other genes, you see areas where they are conserved, and then you make primers based on those conserved areas. Using that approach, nobody could find leptin in anything other than a mammal for 13 years. And lots of people were trying because they wanted to do things like try to manipulate the fat content of a chicken, for example, for consumption. And this would be, messing with that gene would be a way of doing it. So there's a lot of motivation to figure it out, not just science motivation, but also economic motivation. Nobody could find it for the longest time. And then when they finally found it, what they did was use something called gene synteny, which is the sequence of the gene is not conserved, but the order of the genes on the chromosome is conserved. So, and that's the case for leptin. So if we look at where leptin gene is in humans, it's surrounded by this gene here called ARF, another gene called RBM22. And when you use these genes, which are highly conserved, and you look for those genes in other species, like zebrafish, or ultimately Dr. Duff's lab figured this out in birds, you can find these genes, and then you say, okay, what's in between? And what's in between should be leptin, if it's similar to how it is in humans. And that's ultimately how leptin was found in other species, by gene synteny, which is the order of genes which is conserved, and not the sequence of the gene. So if you actually look at the sequence of leptin itself, it's not very highly conserved. Even though its three-dimensional shape, its tertiary structure is highly conserved, the amino acids that go into that shape are not highly conserved. Again, it's a funny little protein. Okay, so I've been talking to you about leptin, but I don't want to give you the impression that leptin is the be-all and end-all that control appetite. So in those 50,000 plus studies that have been going on since 1994 when leptin discovered all kinds of other peptide hormones, proteins that control appetite. So things that can make your appetite go up include neuropeptide Y, melanin concentrating hormone, ghrelin, orexin, PYY, noradrenaline, agouti, ghrelin, AMPK. Things that decrease your appetite include CART, CCK, CRH, MSH, insulin, bombesin, uricort, and so on. Agouti is a really cool. I'll talk more about agouti, I guess, in a different lecture. It's a very cool system because it's not whether or not you have the gene or how much you have of the gene, although that also makes a difference, but whether or not it's unmethylated. And it controls coat color in addition to appetite. So if it's unmethylated, it's always making melanin concentrating hormone, and appetite increases, and also causes this agouti color red hair. There are just a few humans who have this mutation. They have bright red hair, and they have uncontrolled appetites, obesity. Now that does not mean that if you have red hair that you have unmethylated agouti. It doesn't mean that at all. But it just so happens that people with this problem also have red hair, and it is related to their unmethylated agouti. There's also a invertebrate version of leptin called UPD, and this is the only invertebrate version of leptin that I've seen, and I haven't seen anything since 2012 about this version. Okay. So my lab is interested in leptin in terms of where did it come from, what's its evolutionary history, and how does it affect the life histories of non-mammals. So the idea sort of like is if you're trying to figure out how this thing works and it does all these different things, then you can learn a lot by studying the history of this thing. Sort of like if you want to know how brakes work in a Tesla, you can learn a lot by studying brakes of a Model T, for example. All right. So we have done things where we used morpholino oligonucleotides to inject into fish and prevent them from expressing leptin for the first few days of their life, and it dramatically alters their phenotype in the first few days of the life. This is in zebrafish. We have done CRISPR experiments where we're knocking out leptin, get less dramatic phenotypes, but you can see the yolk here is much bigger in the CRISPR fish than it is in the wild-type fish. Notice also these are called otoliths. These things are bones in their inner ear, smaller in the CRISPR fish, larger in the wild-type fish. In the fish where we knocked down leptin using morpholinos, they could not make an otic vesicle. They could make an inner ear, and we got a lot of press for that sort of thing, and because that was supposedly novel. But then when they went back and looked at these OB mice, these guys here, found out that as adults, these guys are deaf, and just nobody thought that it could be related. And so that's an example of how studying a fish can tell you something about what's going on in mammals. We've done with Dr. Jeremy Prokop, who's a former student in this class, actually. from a long time ago. He's now a faculty member at Michigan State. He's done modeling of the leptin receptor and in silico, and he predicted that leptin receptor binds to the leptin ligand in a 2-to-2 conformation, so 2 leptin proteins bind to 2 leptin receptors for active dimerization of the JAK-STAT receptor. We've also found out that leptin does not seem to be following this adipostat model as it does in mammals, in fish. So in fish can go a long time without eating, at least cold-bodied fishes. So not necessarily, I'm not saying don't feed your aquarium fish. I'm saying the fish that live out there in the real world of Ohio, they're just starting to warm up this spring. They haven't been eating all winter long, and they're just fine. These are examples of fish from Arctic char. These are fish that don't eat for several months up in the north, and as they don't eat for several months, the fat in their livers goes down. But curiously, as their liver fat is going down, leptin is actually going up in these fish. Now, that's not what you would predict from the adipostat model, and so the adipostat model doesn't necessarily work for all vertebrates. So we have done CRISPR stuff in my lab with leptin. We weren't the first lab to do it. There was this guy, Max Michel, was the first lab that did it. He published in 2016. We were working at it at that time, but he scooped us, got to it first. But then he sent us the fish, and he's been a collaborator since then. So it was a good outcome there. We have these fish. They are weird. My graduate students are working on these fish and trying to see what other aspects of their phenotype changes by not being able to make leptin. And then Hope Ball, Dr. Hope Ball, is at Neomed now. She did her PhD with Dr. Duff and I on leptin in whales. And the question there was, again, centered on that adipostat model. So whales, bowhead whales in particular, are the fattest mammals, up to 50% body fat by weight. And so if the adipostat model works, then they should be just constantly flooded with a leptin signal all the time, and they should never have any appetite. Just to give you an idea, these are jaw bones from these bowhead whales, and this is Hope in Barrow, Alaska. So what Hope did was she looked at blubber samples from these whales over the whole course of a year, and she found that that they, first of all, they do make lots of leptin. We thought maybe they didn't make any because the signal, again, would be so overwhelming that they might just shut it off, but that's not true. They make lots of leptin, up to, say, 50 times as much as a mouse, for example. But they also have these seasonal patterns where they don't eat. So in the fall, they bulk up and add inches of blubber on the outside of their bodies, and then they spend most of their winter under the ice where they're not feeding or not feeding a lot. When they come back in the spring, their leptin concentrations are really low, and that makes them really, really hungry, really motivated to eat. And they're low even though they still have a lot of body fat on them. And then when they're bulking up in the fall, they have very high concentrations of leptin. So like has been seen in hibernating animals like bears, it seems like whales can adjust their sensitivity to leptin, and we think it's through that protein endospannin, and we hope to add more evidence to that with future studies. This is a slide that shows all the possible ways to affect appetite, and everything that is in a yellow box is a drug that has been or is currently under clinical trial as a mechanism to pharmacologically change appetite in humans and change fat storage in humans. As you know, just by being a human on this planet, there is no magic pill. You can't just go to the doctor and say, I need a prescription for blah, and then you return to a normal body weight. It's a very difficult problem, and part of why it's a difficult problem is that there are multiple redundant systems that have evolved to make sure that you have enough calories. And it's hard to mess with something as fundamental as eating. We evolved under a system where the environment did not have as many calories easily available as our current system that we're living in now. And so we have all kinds of brain rewards that set us up for seeking out high calorie, reward inducing foods. And later on in the semester, I'll show you why, if you're not happy with your current weight, it's not your fault. You got all kinds of things in your cells working against you. Okay, so that'll be the last one. And it's not a magic pill. There have been clinical trials with leptin, giving people leptin injections, and people did lose weight, but it wasn't dramatic weight loss. There's been all kinds of other versions of leptin used in clinical trials that have failed. There's been some use of leptin to help people conceive. I told you about the thing about really low body weight mice lose their reproductive function. The same thing happens with female athletes that below a certain body fat percentages, they will stop having their periods, and leptin can reverse that. There's some evidence that women who are having trouble conceiving, who are also very low body weight, benefit from leptin in that they have an easier time conceiving. But leptin, in terms of a therapy, has not panned out. That Amgen, that same company that makes EPO, has the rights to use leptin as a therapy. And 16 years after its discovery, no, is that right? 26 years after its discovery now, there are no leptin-based therapies that I know of that are commonplace. But there's a lot of stuff out there and more to learn about obesity later on in the class, so stay tuned.